Welcome, Dr. Raj. Thank you so much for coming today. If you could give us the university and college you teach and also an explanation of your field, that would be good.
Sure. So I am an assistant professor at the University of Pennsylvania at the Perelman School of Medicine. And I study brain development.
I see. That's great. So how long have you been a professor?
I started pretty recently. I joined in my position in May of this year, May 2021.
Oh, wow, nice.
Yeah. So I
So
just started my lab and in the middle of the pandemic and getting it up and running.
I see, that's great. So very recent. So to start off, do you have any like outside hobbies or interests?
Yeah, I am a pretty serious outdoor enthusiast. So I really like spending time hiking, camping, skiing, and rock climbing and biking. I would say those are the major things that I like to do. And I also enjoy dance. So I've done hip hop for many years and dance hall. So I would say those sort of round up, and I enjoy cooking. So I would say those round up pretty much my hobbies.
I see. That's great. All right. So let's start out with this. So what led you into your field of study and where do you think it's headed towards in the future?
Um, so yeah, my interest is in brain development. And I think the major reason for me to get into it is because the brain is so complex and there are so many questions that you can ask. And I feel that the more people you have working on it, the greater the chances of figuring stuff out. And so I was really just sort of drawn to the innate complexity of the brain and trying to understand how it develops from, a very small number of cells early during embryonic development to such a complex structure that governs our thoughts and feelings and emotions. So trying to understand the molecular basis of how this complexity is encoded in our genome and how that plays out as the organism is developing.
I see. That's great. Could you give us like a gist of your educational pathway?
Yeah, so I grew up in Abu Dhabi, so I finished high school there, and then I moved to Toronto to do my undergrad. And so I was at the University of Toronto, and I did a bachelor's of science in molecular genetics. And after that, I did my PhD, a graduate school, also at the University of Toronto. And then I moved to Cambridge, Massachusetts, just Cambridge, Boston. And I was a postdoc at Harvard University for 5 or 6 years. And then I moved to Penn to start my own lab.
That's great. Well, thank you for that. So I know you just recently joined, I think you said May as a professor. So did you have you done by any chance any research so far?
Yeah, I started my own research in my lab. It's a sort of continuation of what I was doing as a postdoc at Harvard. And I work with zebrafish, so the model system that I use instead of mice, which would be a mammalian system, I use zebrafish, which is a vertebrate system and slightly less complex than mice and easier to handle. Yeah, so I started working with that, and right now my lab is set up. I have a technician in my lab, and we're starting to do some experiments to try to understand how signaling in the brain occurs during development.
So that's great. So I just wanted to like this is kind, I think I already asked this, like the deep question in research, like, are there any like specific questions you're answering in your research, and also if there's any like data or latest findings in your lab?
Yeah, so the types of questions that I'm trying to ask is determine how cells make decisions during development. So essentially you have early on in the embryo, you have a small number of cells that will eventually give rise to a lot of other cells, more specialized, more advanced cells. And so what I'm trying to do is to figure out this trajectory of how the cells go from a limited number of low diversity cells to a high number of highly complex cells. And so I'm trying to determine, for example, what are the gene expression changes that occur as cells mature from the embryonic state to older stages, juvenile and adults. And then also try to address, for example, how many different types of cells are present in the brain. So can we use molecular signatures, for example, gene expression profiles, to determine how many different types of cells we can delineate in various stages of the growing brain. And then also trying to determine what kind of signaling pathways the cells respond to during development and how these pathways can affect what type of cell the cells eventually become. So essentially trying to tease apart all the molecular inputs that help to shape how a brain cell develops.
Mm-hmm. So like, how do the cells of the formation of an embryo or the brain?
Specifically the brain. I'm following up on the limited number of cells that would give rise to the brain within the embryo. Yeah.
Oh, okay. Yeah. So, do you have any data on that so far?
Yeah, we've published a couple of papers looking at characterizing the different types of cells in the brain of the zebrafish at multiple stages of development. So starting from really early on in the fish development, like 12 hours post-fertilization, which corresponds to a stage where the fish, has a very rudimentary structure, like it just has like a very primitive brain, maybe a beating heart. So starting from that and going up to 2 weeks post-fertilization. So at this point, the fish become larval fish. So they can swim, they can eat, they can start displaying some sort of complex behavior like hunting. And so those fish, we're basically trying to track how the cells increase in diversity from very early on, 12 hours, to much later at 15 days post-fertilization. And what we find is that the cells, you start off with a group of stem cell-like cells in the brain that will give rise to many different other types of cells. And we're using molecular profiles of these cells to determine at what points during development are there bursts of neurogenesis, or the generation of neurons. And we find that, it starts off with about, I think like about 25 different cell types that we can define early on. And we end up with something like 100 different cell types. These are conservative estimates. It's likely that there are more cells than that, but at least molecularly, we can determine that there's about 100 different cell types by 2 weeks post-fertilization. And this number might continue to increase as the fish gets more, develops more, becomes more complex, and eventually becomes an adult.
So how do these, like you say, cells, right? How does the development of like the mass production of like these brain cells, or do they occur in stages? Like let's say like cell proliferation, right? So what are the stages that happen that causes then produces the end result?
Yeah, so generally what would happen is you'll have what we call progenitor cells. So these could be stem cells, or they could be a little bit more differentiated than pure stem cells, but they haven't committed to becoming the final cell that they will become. So they still have plasticity, and they could become a variety of different types of cells. So generally you'll have these group of progenitors cells, and they will, during development, they will divide. So they will undergo cell proliferation, and their numbers will increase. And then eventually they will receive some form of a signal from their environment. And that actually then triggers the next step, which would be cell differentiation. So after they've proliferated and increased the numbers, some of the cells within this pool of progenitors will start to differentiate, which basically means they're starting to go down the path to give rise to the final or terminal cell type that they will become. And then that cell type is permanent. That's it. Like the cells no longer undergo any more division. That's the final cell type. That's how it stays until the cell eventually dies. So that's a general stage or stages. And during development, these stages occur continuously. So these waves of proliferation and then differentiation and maturation continue to happen as the animal is developing.
So do you study like the cells forming the entire brain or specific parts? Because I know there are a few parts in the brain that form first before the other ones. And I'm just curious on how that all comes together, and like how the neurons and like the cortex, they all come together. I know that's like a long process over, it goes from the fetus, embryo, and all that stuff. So I was just wondering, like, how do they come together and what forms first?
Yeah, so I study the entire brain. So I'm not focusing on any one particular region, and just looking at generally what are the principles of brain development. And working with zebrafish actually is an advantage in this way, because the brain is smaller and has fewer cells than, for example, the mouse brain or the human brain. It's actually feasible to be able to study this at the whole brain level in a fish or in a vertebrate system. So that's one of the reasons why I was working with zebrafish to address these types of questions. And yeah, so the different regions of the brain will expand at different rates than other regions. So some regions might grow faster at a particular stage, and then the other region will then grow at a later stage. And those are the types of questions that I'm actually interested in addressing as well. And some initial data that I had suggested that the forebrain, which would in humans, for example, correspond to the cortex. The fish don't have a cortex, but humans would. But the forebrain in the fish is sort of like a primitive representation of the human cortex. We do notice the specialization in the forebrain occurs earlier than in some other regions of the brain. So that could, I would have to do a bit more detailed study, but at least initial data suggested that might be the case. And that different regions of the brain would increase in the neuronal diversity of cells at different time points during development. But that is unclear, like what is the order of things in the brain. It's roughly known, but not with great detail.
I see. So does the rate on how fast or slow it goes, is it based on genetics or is it based on individuals? Like,
yeah, that's a good question. I think in general, it would be dependent on genetics. So the environment of the cells and the type of signaling that they receive at specific time points during development. I think these are sort of hardwired and they would trigger at specific time during development. So it would be a repetitive or recurrent thing. But there could be some individual cases too where, you know, the environment of the individual could affect the level of neurogenesis that occurs. And this, for example, it's not something that I work on, but there are other researchers that are looking into this. But the behavior or the environment of the fish can affect the level of neurogenesis that occurs in the brain. So for example, if you place a fish in a much smaller, like a smaller dish, so that they're sort of constricted as larvae, they don't have as much space to swim around, they actually stop undergoing neurogenesis, or at least decrease the level of the number of cells that are generated in their brain, and they end up with slightly smaller brains. But then if you put them in a bigger dish, they will then readjust and then catch up or like start growing again. So there can be things like this where sort of the stimuli that they receive, the environment that they're in, can affect the level and the rate of neurogenesis that's occurring that is independent of regularly scheduled developmental cues.
so does this neurogenesis process like happen with adults too, or is it just when?
Yeah, so that's like a really important question, and really a lot of work, a lot of research goes into that. So in humans, there is very limited neurogenesis, which is the generation of new neurons in the adult human brain. So far, there have been 2 regions in the brain where people have found stem cells, and they show very limited amounts of adult neurogenesis. In zebrafish, however, this is like another reason why this model system is very interesting to look at. Zebrafish have undergone neurogenesis throughout their entire life. So even as adults, old adults, they will still generate new neurons. And it's one of the reasons why people like to study zebrafish, because not only do they undergo adult neurogenesis, they can also repair their brains. So if you introduce trauma to the zebrafish brain, they will regenerate their brains, or like the section that was disturbed. So they're a great model system to study, stem cells in adults, as well as what happens after injury and how you might be able to induce neurogenesis and repair and things like that.
Mm-hmm. So you mentioned something about like the brain shrinking, and that connection to neurogenesis. So I was wondering if that happens in adults as well, if like any environmental or external factors causes like the brain to shrink. Is that impossible, or is that?
Yeah, so I don't think that the brain shrinks. I think what happens is that the brain doesn't grow anymore. So the thing with zebrafish, for example, is that they continue to grow, as in their body axis increases in size during like their whole life. So if you actually feed them a lot, they will just keep on increasing in size. Obviously, there's a limit, but for the most part, they will increase. So what I think what happens is if the environment changes, they stop growing. So whatever stage they reached, they will stop there, and that's sort of where they pause. But I don't think they'll shrink. So they won't become smaller. It's just that they're not getting bigger. Yeah.
I see. So are there any like misconceptions in your like field of study?
Um, yeah. So I think the biggest misconception might be that people think, oh, why would you study like the fish brain, for example? And I would argue that the processes that I'm studying are actually very well evolutionarily conserved. So many of the very, very early developmental cues that embryos receive are are the same between like fish and humans. Obviously, the genes are slightly different. The genetic sequence is slightly different, but overall, the genes are conserved from fish to humans. And zebrafish share about, I think, 75% genetic sequence with humans. So they're actually a pretty well, like a really good model system to study these types of questions. And so, yeah, I'd say that, just because it's fish and it looks different doesn't actually mean that it's not a great system to study human processes that are relevant to humans.
I see. So has there anything that you ever disagreed with in terms of research and also since you're in the field of science?
So do you mean like, has there been some research that's relevant to me that I didn't think was correct?
Yeah.
No, I don't think I've come across that so far. I'm still early on in my
Yeah.
independent research, so I'm learning as I'm going too. So I haven't come across anything yet where I felt like a study that was published was, saying things that contradicted my own findings, for example. Yeah.
I see. All right, that's great. And to sum up all my questions, I have one last question, which is who or what was your inspiration to pursue your career? And also, any advice for younger people who are aspiring to be in a field similar to yours?
I'd say my inspiration would, it would be my family. I come from a family of like well-educated people, and my parents are both in medicine, they're doctors. So I always knew that I wanted to do something related to either the medical field or biology in general. And I decided that I didn't really want to go into med school. So for me, like research just seemed the next best thing. And this was always my focus. Like once I decided to get into research, I didn't consider any other options. Like I knew that I wanted to become a professor. I wanted to have my own research lab, and I sort of structured my career towards getting that. So yeah, so for younger people, you don't have to know everything. I feel sometimes people are like, oh, do you have to know that you want to become this or that? And it's okay to kind of explore what you like to do. There are actually quite a lot of options now. So you could go into med school, you could go into graduate school, you could do an MD PhD program where you do med school, but also get to do research on the side as part of your degree. So, or you could, of course, go into industry. There are a lot of startup biotech startups that are like taking over many of the large cities. And so and people get really well paid. So depending on whether you'd like to do more schooling or you would actually like to get into the job market after you finish college, there are actually a lot of options, more so than, for example, when I was starting out in my career. So I would say, yeah, just research what you like, what interests you, start talking to people. And if you can go to any kind of career fairs or any kind of information session where people from different fields come and talk about their experiences, you can just listen to one of those things. Usually a lot of the colleges and organize things like that. So yeah, I'd just say, follow your heart.
Yeah, thank you. I think that's great advice. That pretty much sums up all the questions I have. Would you like to add anything last minute?
No, I think, yeah, I think I've mentioned pretty much everything that I'd like to say.
All right, that sounds great. Thank you so much for coming today. I appreciate your time. I hope you have a good day today.
Thank you. Thank you. Thank you for inviting me.
Yeah, of course. Bye.
Bye.